So, good afternoon. Now about the truth in forensics. I see you're eagerly waiting to learn how to commit the perfect crime here. And probably there is some information for you, if I'm not forgetting my pointer here. Forensics is about technology. Forensics is about interpreting genetic data and conveying this information to people who have no understanding of genetics, like people from the court. But forensic genetics also is about individual cases. And what I would like to do now is take you on a tour of those exemplar cases that shaped forensic genetics and the developments in the past years. It all started more than 100 years ago with the development of the classic ABO blood group system, on which the serological methods based that were used until the 90s of the last century in order to perform this kind of work. And it was replaced by DNA fingerprinting, as we all know. Sir Alex Jeffries was inventing this method based on RFLP, where he found out that these fingerprints are identical in humans. And it was also his laboratory who performed the first criminal case. It was actually two cases. It was two rape and murder cases. They had an identical modus of operandi. And what they showed in that case, and the case is particularly interesting because it has a couple of features which we now see in many cases. It was the first time that it was shown that you can extract DNA from old stains. So far, this was only possible from fresh material. And also the authors were looking at different kinds of stains. So they were able to separate the semen from the remaining cells because they wanted to establish a male profile. There was another suspect who was exonerated when they found out that his DNA profile wouldn't match the one from the crime, from the semen. And in the search for the true perpetrator, they were performing a mass test, which means they were inviting more than 5,000 volunteers to donate a sample, type those individuals, and they didn't find any match to the semen stain. Now, the true perpetrator came to their attention because later an eyewitness was hearing when he was talking about how he escaped that mass screen. So the police were asking him, he needed to give a sample that matched the semen sample, and this is how he was found guilty. With PCR and microsatellites, the forensic genetic field was seeing a revolution. Because with this amplification process, the sensitivity was increased by 3 orders of magnitude, and the number of samples that would be amenable for a forensic test exploded. Today, we are using systems that we refer to as STR multiplexes, and they can be used to simultaneously amplify up to 30 STRs, which are independent from each other, from as little as 15 cells. If you smoke a cigarette, you lose about 160,000 cells on average, so don't leave your cigarette butt at the crime scene. With an astronomical statistical value, the random chance of finding such a DNA profile again in the world population is extremely minimal. Now, the interpretation of these DNA data is pretty straightforward when we talk about criminal cases. If the DNA profile from a crime scene sample does not match the one of a perpetrator, then this person, or of a suspect, I should say, then this person needs to be excluded as having contributed this sample. If, however, the two samples are identical to each other, or the DNA profiles are identical to each other, then this person cannot be excluded from donating the sample, and this is what the police can use as evidence in order to solve a case. In the meantime, we have a pretty good understanding on the success rate of these different crime scene stains because we have been analyzing so much. So we can give an expectation how successful it might be to analyze a certain specimen, which is information that is very helpful for the police in order to triage the investigations in a particular case. Now, in those cases where the police do not have a suspect, but they have a DNA profile and they know what it means from a statistical point, they can store these DNA profiles in so-called databases. And here are a couple of examples of European countries, and you see the number of database samples they have from stain samples. Now they also feed in DNA profiles from either suspects or convicted offenders for certain crimes. And what is interesting is to look at the number of stains per individual, because that gives us something like the success rate of such a database. And what we see is that every second to every fifth case, for example, from an unsolved case, leads to a match in the database which the police can use to reopen a case. And that is happening quite often, more often than you probably think. In Austria alone, this is happening 200 times a week. In Germany, probably 2,000 times a week. So with these intelligence databases, police are accumulating evidence for new investigative leads in order to identify crime. Those databases are managed under national law, which is very different in the different countries in Europe, but they can speak with each other. And there are a couple of different contracts and legal agreements that allow for this, such as the PRIM database network in Europe. And if we look at that in a global basis, we see more and more countries establishing those databases because they are so successful and because they help combating crime in a very efficient way. Now let's move from criminals to historic individuals. We can use exactly the same laboratory scheme to evaluate the authenticity of human remains, and people apparently like to do that. One of the most promising, or one of the most known cases to date, and it's also one of the earliest cases, is the identification of the Russian Tsar family Romanov, which came in two different parts. Part one was when they were found a mass grave with skeletal remains attributed to 2 adults and 3 kids, plus skeletal remains of which were supposed to be the servants of the family. At these early times, they were using STRs, but only to describe the samples, not to perform statistical measurements or evidence. This was all based on mitochondrial DNA, which is maternally inherited, and they were able to base the maternal DNA, the mitochondrial DNA of the Tsar and of his wife on living relatives of the 2 branches. However, the statistical values were pretty low because of the mode of inheritance of that marker. In the second part, 14 years later, when very close to the first mass grave, they found another grave with the skeletal remains of 2 infants, forensic genetics was much more evolved. So we were able to type autosomal STRs as well as mitochondrial DNA as well as Y chromosomal STRs, which you can use in order to test paternal lineage. And it came up with a cumulative likelihood ratio, which is astronomically high. What you usually have at court, if you have an LR of 1,000, that is considered to provide strong evidence in favor of the first hypothesis, which is usually, yes, this is the person or this is the father of this person. And by this value, there are no doubts that the Romanov family is identified and the case can be closed. Now, haploid markers are quite interesting in forensics because they allow us to look in the past. And they are, because both are uniparentally inherited, we can use them to look at people or remains of people that lived in the more distant past, where we could not use autosomal markers that are recombining in order to capture that signal. One example is King Richard III, who lived more than 520 years ago. And the beauty of these royal families is that they have a very well-documented pedigree that you can use in order to try to find individuals that are living today to provide a sample. Happened for 2 individuals from the maternal lineage, which comes from the sister of the king, Anne of York. And for the paternal lineage, where they needed to go back to his great-grandfather Edmund III, down the pedigree to 5 living individuals. Here are the results. The mitochondrial DNA genome from the skeletal remains was identical to the 2 of the living relatives, with the exception of 1 base in those 16,569 bases that can be explained with an evolutionary hotspot mutation. However, The Y chromosomal results didn't fit at all. The skeletal remains have different Y chromosomes than the 5 males, and the 5 males that should be related to each other according to the pedigree have a different Y chromosome. So how can we explain that? Is King Richard III's remains authentic or not? Louis was talking about the first and second sexual revolution, and I'm making a strong point here that at least the royal families had sexual revolutions in the past as well, because what we can consider and what we need to consider looking at these 5 individuals, that there is false paternity. A conservative rate would be 1.5%. So on average, 1.5% of the men think they are the father of a child, and in fact they are not. You guys here are too small of a group to statistically be a false father, so you are off the hook in that respect. But the way how we can weigh this information in forensics is that we try to combine all the evidence and try to bring it up into a likelihood ratio, including the factor of false paternity. And still, the evidence is overwhelming for the hypothesis that the remains belong to King Richard III. Those haploid markers in forensic genetics are curated by databases. We have 2 databases, MPOP for mitochondrial DNA, YHRD for Y chromosomal work, and the research groups, they are forming around these databases, and we use them in order to provide a databases for frequency estimates. But what these databases also do, they do a quality control of the data. And in forensic genetics, we are extremely sensitive to quality control. You don't want to hear the word error in court. That's something you don't want to say if you stand at court. And this is why we have been looking into tools in order to develop high quality data. And what you need to do is, you need to do a posteriori checks on your data. Since they are both inherited along a phylogeny, we can use phylogenetic tools to test the quality of the data, for example, with quasi-median networks, who we can use to display a table in a graph. Because our eye is not used to find errors in a table, we just don't see them. But when we turn the table into a graph, then our eyes can more easier spot errors. And what you see here is a typical phenomenon. A sample becomes an outlier of a data set if it's phylogenetic distance or if it's an error. Now, you can use this tool and look at all the published data in the literature, and you find that more than 80% of the population studies published in medical genetics, population genetics, and forensic genetics contain errors. It stopped in forensic genetics in 2010 when we started to apply the rule, because those papers cannot be submitted to the journals anymore. But if you look into Science and Nature, we sometimes find error rates up to 100%, which means every single sequence that is published there is wrong. So the forensic genetic field needs to have recommendations and guidelines in order to be able that we are as error-free as possible. And one reason for this is, if we have errors, that is lowering the frequency of a haplotype. And that means that we put the suspect at a disadvantage, and we are not supposed to put a suspect at a disadvantage. And this is why we need to care about the quality so much. Forensic field is moving into sequencing of the entire mitogenome. We have a couple of different strategies depending on the degradation of the DNA at hand. I would like to draw your attention to the micro strategy here that can sequence very short fragments of mitochondrial DNA and bring this in a connection to a case. That goes back to the year 2014, where 43 male students went missing and haven't shown up since then. And there was strong belief that they were killed and burned. We received a bag that they found in the river, and that contained ash and 17 individual, very small pieces of severely burned and degraded specimens. These are the typical hopeless cases where usually you have no success. You try your best, but you don't get anything out of it. We were pretty lucky because one of those specimens, after removing the ash, shield around the item, we were able to find some tissue that contained enough nuclear DNA to do a conservative STR capillary electrophoresis genotype. So once we set up the genotype, we were getting the information of the 43 families. We got 42 mismatches, and one family matched the DNA profile, and the likelihood ratio was far above 1 million. So this was a true identification of one of the remains. But the other 16 gave nothing, not even a single signal with real-time PCR. Now, the question that we ask ourselves is, if there is DNA present in those remains, then the only way how it can be present is in very highly degraded way. And we can artificially reconstruct this by a DNase experiment here, where we see that the critical value of 1,000, this is the copy number which we don't like to go below that in order to get authentic results. With 143 base pairs, we reach that after 8 minutes of incubation with DNase. And with 50 base pairs, it takes 1 hour. Or if we look at that in a more natural degradation process and we look at remains of the medieval times, then we see that exactly the same extracts contain almost no DNA worth of 143 base pairs, but more than 20,000 copies of 50 base pairs. So we're not talking about low copy number here. We are talking about size. Now, we can't use PCR to amplify those, but we can use different strategies, and we were looking at primary extinction capture that was developed for sequencing the Neanderthal mitogenome. We brought that protocol into a forensic context with all the negative controls and positive controls that are required, and were, after 1 year of validation work, using the remains from Mexico, 9 of those 16 gave results, which to me already suggests this is the way to go in forensics with heavily degraded material. 2 of those were human-specific and 7 of those were animal-specific. So that doesn't change the fact that the method is superior. We just recently published this in Genes. So we arrived in MPS, of course, because you can do this kind of work with capillary. Now the question is, what else? Where else is forensics moving in terms of MPS? Now we can discriminate between identical twins, because we know from statistical considerations that identical twins differ by 1.5 bases on average in 3.2 billion bases or what have you. You can't find those with capillary, but you can find with MPS, and that has been demonstrated a couple of years ago. We are also start to sequence those STRs. So far we have been looking at the fragment size, but we have some of the STR markers where you can explain the variation, but we explain only 50% of the variation with size, and the remaining 50% goes back to the sequence. So we can increase the discrimination rate, which is probably not necessary for a full profile, but it can become necessary for a partial profile or for a mixture profile, which have completely different statistical outcomes. As you see, those sequences down there from allele 15 are all the same size, but they have a different sequence. On the right side, you see a couple of EU projects and US projects that are dealing with this kind of work. So what is the future in forensic genetics? We certainly reached the sophistication stage. So we try to sequence all our markers, which is the best that we can do in order to arrive at the genetic information. If you have this, you can get more if you look at genetics than the sequence of a marker. And we are exploring that in different ways. But also the field is experiencing a paradigm shift in the way how DNA is used. So far, I have been only talking so far about identification, which means comparing DNA profiles between 2 samples from the person or between samples from persons who are related in the same pedigree. But if we can also use DNA to predict features. And the application are those cases where we have a crime scene sample that provides DNA, we have no suspect. We put the DNA sample on the database in order to try to find a suspect. This is not successful as well. So we have an unsolved case that sits on the database. What can we provide in order to give investigative leads on that case? Can DNA do something about here? And of course, you know some of this work that has been done here, particularly in the Netherlands, Rotterdam, Manfred Kaiser, he has been looking at pigmentation, eye, hair, and skin color, and he has provided a lot of pioneering work in terms of trying to predicting those. I'm not agreeing to what was discussed earlier about facial recognition. I don't have the time to talk about it now, but maybe we can talk about it in the break or in the question section. We have pretty good models and markers to predict ancestry. And that has been shown by our colleagues from Santiago de Compostela, Chris Phillips and his group, already in 2004, when they were typing the DNA of an unexploded device of one of those Madrid bombing devices. Now, the police was finding the terrorists with conventional means, but the DNA analysis at that time would have pointed at the right geographical origin to search for those people. A lot of forensic genetic research programs in the past has been looking at ancestry informative markers. Here is one called Eurofortune that just ended last year in order to improve the prediction and the association between the DNA and the actual geographic location of these individuals. Finally, I would like to briefly talk about chronological age. Age is forensically important in different means. It has been established that hypomethylation in general, but also hypermethylation of some CpG islands is strongly correlated with age. Now, we cannot perform epigenome-wide scans on forensic samples because they do not contain enough DNA. However, a couple of our groups have started to look at carefully selected sets of markers. These are few markers with very strong correlation to age that we can even use on samples that contain less DNA than those with the epigenome-wide studies. And so far, we have reached a value of about plus minus 3 to 4 years of an error, which is probably as good as eyewitness or even better than eyewitness because it has the advantage of having a statistical fundament that comes with the result, which we don't have when we think about an eyewitness. I would like to mention a EU Age 2020 research project that just started a couple of months ago, Visage, where we are exactly looking at this prediction of pigmentation, ancestry, and age, and try to optimize those systems. A lot of ethics discussion is involved here, which is extremely important, but still, the technology is not at its final stage. We need to invest more here. And as I mentioned before, the societal impact and the ethic dimensions of the work that we do with databasing, but also with prediction work, has so far been underestimated. And it's now the time when these people are involved, when social scientists are included in these discussions, to perform research in order to shed more light into this. With that, I come to the end. I would like to acknowledge a couple of funding bodies in Europe, Austria, and America that finance the work that we are doing. I'm having a brilliant team in Innsbruck that I'm very happy to work with. I'm thanking Thermo for having me here, and I thank you for your attention.